you all for coming back in good time. So from this session, we have our friends who will be taking shots of filming this session onwards. So if you need someone to do a makeup for you, I volunteer. You can get some powder and just do some touch up for you if you want. Otherwise, let's take Harry's talk.
Okay, thank you. Since everything is going to be recorded from now on, especially questions from the audience and from the chair, I will be very diplomatic, and I'm not going to criticize EFSA anymore for whatever reasons. Okay, the goal of my presentation is to lead you to the next session, which is on the completeness of the dossier. So what I will do is shortly address, I think, the principal components of a risk assessment dossier. What should be there? Not saying that if everything is there, it's okay and it's approved, but at least you as a risk assessor should pay attention to the issues which are important. And then I'm not going to talk on the details because that will be up in the follow-up session and also tomorrow. John will talk about it, and Jeff will talk about it, and Bob as well, at a certain point in time. So I'm not going into details on how the risk assessment should be done, but I'd like to highlight just a few main issues where you should pay attention to. Is that okay? Well, even if it's not okay, it should be done to inform you on the content of a dossier. And if you want to interrupt me, please go ahead, knowing that everything you say is going to be recorded. Now, this is an overview in a nutshell of the main items or these issues you should look at. First of all, the genetic modification process, methods, and also what is the donor and recipient and what is inserted, genomic aspects, arrangements of what is inserted, gene disruption, potential disruption, I come back to that later, stability. So I call it just genetic modification and related processes. Then a key issue for a dossier is what's there, what is new and what is expressed in a crop, which we haven't seen before. Sometimes there's nothing expressed, there's something deleted or suppressed. So you would like to know what is there or what is not there. Are there other new compounds being expressed? Is it very important? Is there a change in alterations in consisting constituents already? That is important. And is there a change in possibly alterations in metabolisms? The phenotypic, agronomic, and compositional analysis is key. I, as a toxicologist, probably would first go to this section and see what has been provided in terms of data on the composition. Is there any change there? And if so, you could go on further, but this is the key issue. We discussed it a little yesterday, to what extent molecular characterization can help you in the final safety assessment. That is, you'll find out today and also tomorrow. I'm not saying that molecular characterization is not needed, but what is absolutely needed is a good idea on potential compositional changes. Now, with respect to toxicity and allergenicity testing, of course, we are dealing with new proteins, and other possibly new substances, but yeah, toxicity and allergenicity should be looked at. Now, we listened carefully this morning to Bob, and he says, well, new proteins. He didn't say who cares, but in terms of toxicity testing, there is not a great need to look at. I agree on that, with that. Um, the the thing with newly expressed proteins is we need to know where does it come from, what are the sources of the protein, and what is the history of the use, and what is their function. And if an applicant would describe that in details, then I think that would be acceptable. Of course, we want to know something on the degradation behavior of the proteins, either in vivo or in vitro systems, to get an idea whether there is a rapid degradation, yes or no. I remember a couple of years ago, well, at least 10 years ago, we had this discussion, and people in the US may be aware of it, that was related to Cry9C, BT, cry protein, expressed in corn, I think. And that turned out to be, that was a BT protein which was stabilized, wasn't it, Jeff, by one mutation, one single mutation, which made it stable in order to prolong the insecticidal action of that protein. That was the purpose, wasn't it? And we struggled in those days. The product was not on the market yet, but we struggled with the fact that that stable protein did not degrade very quickly in all kinds of simulated stomach conditions. So we had a problem with stability of that protein. Not a problem, but we were faced with a fairly stable protein. And what would you do with that type of protein when it comes to toxicity and allergenicity testing? That is still an open question. So toxicity testing and allergenicity testing is always an issue, depending on the type of protein you're dealing with. And then there are a few other, I would say, additional issues to look at, and I'll come back to that later. Now, this is a list of issues to look at, and if you are going to open a dossier, I don't know, Godwin, that will happen. They will have a look at the dossier, yeah. And I'm sure that you will find these issues here described. And last but not least, I think the risk characterization as such is, as Bob also said this morning, is an essential thing, because that is the chapter or the part of a dossier where everything comes together, and you should come up with a final evaluation of the data you have seen and come to a conclusion. Now, Bob talked already this morning. The content of a dossier is focused on a science-based risk assessment done by the applicant, by the way, not by the regulator. The applicant should do its job and come up with a risk assessment, which then is going to be checked by the regulators. And the principal things we have been discussing at length this morning. Now, these are the issues to look at. It's the characteristic of the donor and the recipient, the genetic modification process, its agronomic and phenotypic and compositional properties, its potential long-term toxicity and allergenicity of new gene products and whole plants. Now, with respect to long-term toxicity studies, yeah, I in principle would say, well, it's a protein. What are you talking about? Do we need to have long-term toxicity studies? No. But if there are alterations in your product, compositional alterations which have led to higher or lower levels of natural constituents, which are outside known ranges, then you may want to do some further toxicity study. I'm not saying necessarily long-term studies, but there is an issue there. There may be an issue there, but it's all up to you to decide when and when not. Nutrition is an important issue. There are some models to check on the nutritional changes. Influence of processing is of course important with GM foods, but also any new foods, and possible changes in dietary intake. If there is a promotion made with the product by saying, well, this has much more vitamins and minerals, so you eat it, people may intend to eat much more than recommended, so there could be a problem in terms of intake, and also with respect to long-term impact. Now, I should not go through all the details when you are going to characterize the donor and the recipient organism. I'm listed here, and I'm sure that this will come back in later talks and in your exercises. The thing on the use of the recipient plant is, what is its role in the diet, and especially what is the history of so-called safe use of your recipient. That is a very important description of how it has been used, what was the population being exposed to it, or are certain segments of the population less or more sensitive to this type of food. So composition of consumption of particular subgroups is also very important to look at. Now the molecular process I would almost skip. I mean, you'll find the data on the way these products have been transformed or transformated. What is their expression levels is very important. Deletion and silencing as well, and the phenotypic and the genetic stability, of course. Yeah, it's stacked transformation events. I put a question mark there. For certain organizations like EFSA, this is an important point for stacking, and for others of less importance. And last but not least, the potential of horizontal gene transfer. Well, this is in particular important when it comes to marker genes, which could create resistance with certain bacteria, and they could interfere with medical treatments. I think by now this issue on marker genes has always been in practice resolved, isn't it? We use a few old markers genes which are very well described and known. There are no new marker genes which have antibiotic resistance profiles anymore, and further on there are some alternatives. So I think this problem faded away by now. The Commission had demanded to ban it completely, which did not happen in practice because we still are having some dossiers where these marker genes are present, but they are very well known and we know the risk of it. If you want to know more about these issues, go to FAO, WHO, and in these famous Bibles, the orange one and the green one, and you'll find more about it. Now, molecular characterization, the expression, gene expression is a key issue, and also changes at the protein level, but also RNA, etc. And and things of gene disruption and the potential for formation of new open reading frames is an issue to look at, and gene silencing as well. Now, the comparative analysis that in terms of phenotypic properties is an important thing, and I've listed here quite a few parameters to look at, which is normally done by the applicant through field experiments to look at the properties phenotypically of the new GM plants. And I think this is a very important issue. We have experience with this chapter on phenotypic characteristics of a plant, and we think, well, they may signal the occurrence of so-called unexpected effects. If they would be there, you might see something in, I don't know, so certain characteristics, pollen viability, flowering time, morphology, the colors could change. So that could give you a signal for well, unexpected, unintended effects. The actual changes are difficult to interpret. There are, in many cases, there are large variations in these values, and you have always to take that into account before making a decision. So it's not just the differences between GM and non-GM, but also what happens in nature with these parameters, depending, of course, on changes in environmental systems. Composition. In various regulatory documents and guidance give indications that if composition has to be dealt with, you need to do you need to do field trials in order to prove the point of other changes ongoing on in the composition. And this is an example of EFSA, but it's only EFSA. This is not a complete template for all regulatory systems to look at when it comes to the performance of field trials. An important thing is the environment should be representative where the plant is grown and also being exported to. You need a number of sites to look at. Over 1 or more years, depends on the number. You could do with 1 year if you have enough sites, otherwise you could spread it over 2 years. If you don't have indications that there are severe environmental changes in the condition. The comparator is important to choose, the non-GM comparator to choose at each site. And you need to include a number of commercial varieties to get an idea on natural variations in the parameters you are going to measure. And this is a very important point. Now the discussion is always how many should I use, or could I use historical data on plants, non-GM plants, go to a database of ILSI, of other organizations, and find there the natural variations. OECD in their consensus document, they have described a number of crops in terms of contents of constituents, and I find that informative, however qualitatively not the best, because these data come from literature and we have no very good information on how these data were generated. If you look at the ILSI database, it's qualitatively much better database, because first of all they use validated methods for analysis, and secondly the crops where the data from were gathered were very well characterized, including the environmental conditions. So if you want to do something with historical data, I would say go to the ILSA database, and maybe OECD second place, but that's my view. Now the composition of parameters sounds logical, and they are well described in details in the OECD consensus documents. The basic idea of having an extensive compositional analysis in the early days when we were looking at GM crops was that let's take as many compounds as possible as we can measure, so to have an idea if something would change unintendedly, we may be able to pick it up earlier. So that's why we have ended up with a broad array of components to be analyzed. But as time goes by, we probably could think of reducing the number of components to look at, because up to now we have seen some changes, but not dramatically due to the genetic modification. And so maybe we have reached the stage where we could say, well, let's select a number of key compounds only and look at possible changes. At the other hand, there's progress in this area of analytical measurements of crop components, and this is all automated. It's in one run, with mass spectrometry and what have you, you could do the whole job then. So it's cheaper as well. So, but this is an issue for OECD to look at. And Jerry, you're going to OECD every now and then, huh?
Yeah.
Give the message, please. Now, in terms of field trials and testing, I want to emphasize that first of all we are looking at differences between the GM and non-GM. Yeah, there is always something which I should have repaired this this slide. The idea is that first of all look at the parameters in your GM crop and compare them with the non-GM, whether there are differences. If there are differences, then you should look at whether the differences would fall within the range of natural constituents. If they would fall, then I would say, well, end of discussion. If they would not fall within the range, then I would not say there is a problem, but you have to look at it further. So be careful. So it's difference testing to identify the difference, a potential difference, and then equivalence testing to see whether it would fall outside or inside the natural range of components. There is something with these slides which I should, which I will repair later on. But that is the philosophy. Now, to get an idea on natural ranges, as I said, you can go to databases, and you can also go to your data on the varieties, commercial varieties you have included in your assessment. So sometimes you use 6 or even more to get an idea on the potential changes in composition. What is very important in the analysis of your compositional data is, which components have been changed? Is it consistent throughout all the field trials done in different countries under different climatic conditions? What about the natural variation? Dose dependency is not a point here for compositional analysis. So, but look at these natural variations, and then, as I said, you can refer to data from reference lines, but also literature data, and OECD and ILSI. But the practice is, I don't know how much experience you have in this field, but you find always differences with any GMO versus a non-GMO in terms of composition, or also phenotypic alteration. There's always something there. But the thing is it a matter, is it consistent? Are it certain compounds which are related in certain physiological pathways which are affected or not? Or is it just a matter of changes by chance? So even a statistical difference between a GM and a non-GM not necessarily means a potential health effect. It depends on the type of change. Is it consistent, or is it just by chance? Since you are measuring many parameters, over 100, so you always find something by chance. So be aware of that. And we in EFSA, it becomes a bit less now, but we are always, when we have to defend our opinions and we describe the alterations in the composition carefully and into details, we always have comments, either from member states or environmental groups, and they say, well, you see, there are differences, why didn't you pay attention to it? So the evaluation is a key thing. And since there's always a change in something, there are always people who have their views on that. So this is I would not say it's a source of fun, but either of annoyance. Now the other thing, and I don't know how much time during the compositional analysis you will spend on it, but it's the issue of unintended effects, which could be observed in certain parameters, like changes in enzymes or metabolites, or also changes in in the phenotype. Now there are different ways of approaching the issue of unintended or unexpected effects due to the genetic modification during the compositional analysis. You could follow the approach with the classical, just measure the compounds and see what kind of compounds are changed and to what extent. There is a disadvantage here because it's a biased selection. I mean, you choose the compounds to look at, and you will never look at all the compounds, which is impossible. You can only look at compounds for which an analytical method assists. So there are some disadvantages there. But in the classical sense, it works. I mean, an extensive analysis should give you a pretty good idea on possible changes which you would not expect. The more modern way is the non-targeted profiling methods like transcriptomics and proteomics and metabolomics. There is no time here to go into that too deeply, but I think these are methods for the future. And the methods which are very promising up to now is metabolomics, where you just make I don't know whether you're familiar with it, but what you do is that with one analytical machine or a couple of analytical machines, MS and mass spectrometry, NMR and infrared spectrometry, you can make a picture, I would say, of an extract of a plant and compare the picture of that GM plant with a picture of an extract of a non-GM plant to find differences in patterns. So that's an overall pattern you're getting. And instead of looking at all single compounds, you look at an overall pattern and try to find differences. In the area of metabolomics, of metabolomics, I think that has already been proven to be successful. There are a number of reports in the literature which show it's a fruitful approach. For proteomics, I have my great doubts. Proteomics is very difficult to do and also to interpret potential differences in your patterns. Moreover, I have no idea how much proteins you would extract from a plant. Is it 1% or 2 or 3%? Anyway, it's very little. So I don't know whether that is very representative for what you are trying to do. And transcriptomics is difficult, but not impossible. And there are some good studies in that area which shows you, well, it could be very useful when you would like to extend your compositional analysis using these profiling methods. Now, toxicological assessment, that's all for Bob. And as I said, I mean, it would be good to have an idea on the characteristics of the protein, its function, how it has been used, and how familiar are we with those proteins. And but also the issue of possible alterations in the content of natural constituents is a thing to look at. And then, animal feeding trial with whole food feed is a subject as such. And my position is that don't do these type of experiments, and I will show you tomorrow the limitations of that type of experiments from a toxicological point of view. You're not going to resolve problems by doing these experiments. It may be some kind of reassurance for the consumer, but it's certainly of very low scientific value, if any. And you should consider these experiments only when you have a specific trigger to do it. Otherwise, forget it. But it's sorry.
This is what the EU now recommends for the next phase.
Routine for each GM product. And EFSA has always been contrary to that position. EFSA says in some guidance documents that you may want to do these experiments if you have a trigger to do it. If you are dealing with a GM product which has multiple changes in constituents in the composition, or if you would have a problem with the expression of open reading frames, and you are not sure whether they are expressed yes or no, then you may want to consider those type of experiments. But you better first try and think of other types of experiments which are easier and simpler to do than a feeding trial, because as Bob said also this morning, I mean, the food matrix is tremendously complex, and you have many interfering compounds and components present, so that makes the type of experiment very difficult. I'll show you 2 examples tomorrow of a feeding trial. One which was a complete disaster. I brought it because it was a disaster, and another one which was reasonable, but which did not give much clearer answers than we had already.
Which animals?
Rats. This is, yeah, I'm talking about laboratory animals. It's rats and mice.
Okay.
But, yeah?
But that does not mean that you should not assess for maybe allergenicity, because naturally some people will react to some proteins, normal proteins.
Sorry, I didn't get the question.
Okay, I'm saying, you're saying we should not consider toxicological assessment. Is it just for feeding trials or in general?
You mean this point, feeding trials?
Yeah.
With whole feeds?
No, the comment you made earlier that you should not consider
With proteins?
doing, I don't know, maybe I
Okay,
misunderstood.
so toxicological studies is, if I got you right, can be done, but you don't recommend whole food feeding.
That's right, yeah, sorry for the confusion I have. Yeah, this is, let's say, on proteins or specific substances, and it also on new constituents. If there is a gene expressed and which would change the mineral content or vitamin content or whatever beyond known limits, then you may want to do a toxicity study with that specific compound, if needed, not necessarily always necessary. But this issue on feeding trials is with whole food feeds. Now, the Europe has said, not Europe, the Commission has said, you should do these studies anyway because we are not sure whether your risk assessment is transparent enough and it does not give you enough certainty on your final outcome. That's their reasoning, that they think you have to do it anyway. The truth is that in Europe there are quite a few member states who has always been in favor of doing this type of experiments anyway. And I think that has to do with trying to higher the public acceptance of GM foods in general by saying, well, you see, we have also done animal experiments with the whole food feed and nothing happened. So I think that is an argument to promote animal feeding trials. But my view is you should not promote these studies if you know that the power of these experiments is very low, if any. And it would not, from a scientific point of view, add to your risk assessment. So there's a division here. The sciences says, well, don't do it unless you have a good reason. And the risk managers who say, well, we want it because we could ease and please the consumer. Probably more. Well, yeah, that is a kind of argument I don't like, but this is reality.
Any questions?
Yeah.
Just come in, Tom, you're probably going to talk about this in more detail tomorrow, I imagine, when you go into some of the trials. But also, the other thing that's a problem with animal feeding trials is you can't use a high dose strategy, which most toxicologists want to see to force the issue of whether there's a toxic effect or not. And that's probably, I don't know if that you accept that one, but it seems like a
Yeah, in general, that's true. And how I will show you tomorrow. How would you feed a rat a fresh tomato? How would you do this? So there are some problems there. But your point of high dose, Bob, is a very relevant one, and I'll show you some examples tomorrow. Okay?
Somebody else had a question.
Yeah. Who else? Okay. Now, protein assessment, Bob, that's all yours. There are many ways to do it, or not many, but there are a number of ways to do it. I think sequence homology is one of the best things to look at. And yeah, repeated dose toxicity studies, that's in the EFSA guidance document. If an applicant does not have enough information on the background of the newly expressed protein and its use and potential exposure of humans and animals to that new protein, then an applicant has to consider repeated dose toxicity studies, which in practice are 28 days studies. But I always say I'm not talking regularly to applicants, but I just say, well, do your best. I mean, get as much information as you can on the history and occurrence and use of that protein, and avoid animal experiments. Because I'm really of the type that we should avoid as much as possible any animal experiment. And if there are alternative methods, in vitro methods and in silico methods, use them and not animals. Now, allergenicity is for another expert. He's coming tomorrow or the day after? Rick Goodman?
He's going to be talking tomorrow afternoon.
Tomorrow afternoon on allergenicity. So I skip that. I'm always amazed that biotech food has attracted a lot of attention, in particular from environmental groups and consumer groups, on this issue of allergenicity. And why is that? I mean, we are faced with so many novel foods in general. And, well, allergenicity is always an issue you have to look at. But I can't find a reason to think, well, allergenicity and GM foods is very particular as a subject. But ask Rick about it tomorrow. We know that lots of foods are allergenic for lots of people who are sensitive to it. And of course, you have to look at allergenicity also with the GM foods. And because we know we are dealing here with atopic individuals. And the other big thing of allergenicity is there are a number of test systems, and Rick will tell you more about it, but none of them is foolproof. So you have to look at the results of all these different test systems, serum screening. Also animals can be used, but none of them is decisive. So just take this, what we call comprehensive risk assessment approach. That's both for the newly expressed protein and also when there are changes to be expected in proteins which could be allergenic. Well, this is a scheme, it's the weight of evidence approach developed by FAO, WHO, and this describes the whole thing. The characterization of the source of the gene, is that allergenic, a sequence homology, and what have you. It's all described there. But Rick comes back to that tomorrow. Now, nutritional assessment. I can be short on that. I mean, it is an issue. If there are differences in your GM food, then you have to look at possible consequences from the nutritional point of view. Also in terms of bioavailability, that may have been changed. And yeah, the choice of your model to test for bioavailability, I don't know. That's not an easy issue. But there is experience in this area. When we would deal with GM crops, which have a deliberate change in the nutritional content, then you may want to do something in this area. Yeah, this is the same thing. Now, exposure assessment is crucial. We have a new GM product, so we should know, well, if it's fairly similar to the non-GM, then there is no worry. But if there is a substantial change, then we should do a classical, I would say classical exercise and do the assessment of exposure of people in general, and maybe special segments of the population as well. Intake, of course, is an important thing, and also the other thing is that do we need post-market monitoring? If you have an idea on a new GM product and you are not sure, first of all, on consumption, you may have an expected consumption. If that protein, that maize of rice, has a higher vitamin E content or A content, and people would think, wow, this is a very nice product for me, so I'm going to eat more. So you would like to have a better idea on possible consumption by consumers. And so you may want to organize some kind of post-market monitoring once it's released in the market. That should be done then under controlled conditions, and the applicant has to come up with a model for that. So there may be some reasons to consider post-market monitoring. At the other hand, if it comes to safety, then I would never recommend a post-market monitoring system to check on safety. In other words, don't try the market out when it comes to specific safety issues. You better resolve your safety problem before you release it on the market. But for nutritional aspects, as I said, exposure could be helpful. Now, Bob has talked already this morning of the final point of this whole thing on risk assessment is the risk characterization. I mean, what is the view of the risk assessor, looking at all the data comprehensively, and find out, is the data set complete? Is it complete enough? Or do we need additional data? My experience in this field is we need, in most cases, more data than the applicant has delivered. And whether he has been reluctant in submitting data, I don't know. But there is an issue of, incompleteness in dossiers, and that should be evaluated during the risk characterization. There are a few minor issues, and then I stop, is that, and you should look at this as the risk assessor, and that is you may get information on a new protein with a so-called history of safe use, or the use of a food which has a known history of safe use. And as I said yesterday as well, I can see why, and we are around, and we have been exposed to proteins and foods all the time in our life. So there is some kind of history of safe use. It is very difficult sometimes to quantify it, but applicants should do its best and try to get a better idea on where does it come from, what is its function, biological function, and have people been exposed. The notion that, for instance, this protein comes from E. coli bacteria, and we have been living with bacteria all the time. Yeah, this is true, but if we could get a better idea on exposure, that would be helpful. So a better definition of the history of safe use would be very helpful. And don't use it all the time, and further, no providing data. Canada, as I said, is a country where they spend quite a bit some time on the concept of history of safe use, but this is on novel foods, and it says it characterizes a number of parameters. You should look at the characterization of the source, If so, the compositional analysis in terms of bioactive compounds, etc., toxicological and nutritional information is needed. With respect to consumers, we would like to know, to have better idea on the exposure over generations, the number of consumers, specific consumers group, etc., etc. Go to the Canadian document that's here, and you'll find out there are a number of more detailed aspects to look at in order to decide whether we can use the history of safe use as a concept for our safety assessment of GM crops. And then, you can think of a similar thing when it comes to the characterization of newly expressed proteins. What do we know about the protein, the source of origin, and the use and its function? And this is all to avoid, as much as possible, animal experimentation. Just improve your logic on characterization of compounds you are faced with. And do not do animal experiments because it's in the guidelines. Just by reasoning, try to avoid it. Now, as I said, post-market monitoring can be an issue if we have some doubts on I would say the occurrence of unintended effects during the pre-market assessment. We would like to confirm possible consumption of a GM crop, which may have an altered nutritional composition. And allergenicity could be a point which you would like to try out further under controlled setting and conditions. But as I said, it should not be a substitute for a pre-market safety assessment. Conclusions. I think globally, and this is important, biotechnology is global. It's not a local. Yeah, it may be in certain countries local, if you have some interesting crops there, which you would like to grow, which could be very important for a local market. But in general, I mean, biotechnology and the application in foods is global. So I'm very happy that there is a common consensus on the principles and on how to do the risk assessment. There are differences, let's not forget it, and I mentioned a few, like the strict requirements of the EFSA to assess stacks, and also the routine testing of whole food feeds. But this is not going to be written in stone forever. I mean, I'm sure we will be flexible in that, and the issue of routine testing of whole food feed, depending on the results which are coming now from current running projects, probably will change that position. And the more opposition from the scientific world on this issue, that will be very helpful. But there is a further need to harmonize aspects for risk assessment. Like, for instance, the use of databases, the quality of databases to get a better idea on natural constituents and natural variations in these constituents. Selection of compounds for your, and also phenotypic parameters for your risk assessment we could think of, and the introduction of these more global profiling methods could be very helpful. But there are a number of organizations who should spend time on it. And I think that's about it, yeah. I'd like to thank Claudia Poletti as deputy head of EFSA, the GMO unit. Thank you very much.
Thank you, Harry. Any
Yes,
additional questions there?
yes.
I think I have two questions. The first one is there always, like, for example, published data in UC databases or OECD databases to be able to account for any differences in the GM and the non-GM that are brought about, for example, by natural variation
No, no. I mentioned the database because of to get an idea on natural variations. You have discovered a difference in the calcium content in your GM versus your non-GM, and then you want to see, well, calcium varies all the time because of different environmental or climatic conditions, and you would like to get an idea on the natural variations. And those databases are very useful for it. Now, ILSI has a database, and ILSI puts only data in that database with validated methods. So it's a very good database. OECD refers to the literature, and that is not always of high quality, the literature.
So I can always account for any natural variation by referring to a database. If I'm like a regulator, and I've received this information from an applicant, and I can see there is an obvious difference between the GM and non-GM.
Well, if this were true...
is it like...
no, not always. I mean, it depends on the compound you're looking at. And ILSI has not all the compounds, but and ILSI has crops now, what is it, maize and soya, complete. For the contents in natural constituents. And OECD refers to literature values, which, as I said, is not the best.
So what other sources can I use?
Well, the most, yeah, it depends. If the applicant has done a field trial with GM versus its isogenic counterpart, and then he's included reference varieties, commercial reference varieties, that would be very helpful for you.
Okay, you study
at the same time.
Okay. Then another question is that, okay, yesterday we talked about, or the other day we talked about data transportability. If there is a potato that has been, maybe there's a genetically engineered potato that is growing in Peru, and the agroecological zone is the same like as in southwestern, is the same as in southwestern Uganda. They have similar agroecological zones. So as a regulator, what information would I ask the applicant to provide for me? Because you see, there is already published data.
Yeah, but from South America.
Yes, but maybe I'm just saying, I don't know if it's true, but maybe the agroecological zone is similar between South America and this southwestern. Can I just use the published data, or do I have to ask him to now give me that?
Well, I would probably ask additional data for your country in order to prove this point, to confirm it. You don't have to do these extensive field trials, but you need some kind of confirmation. That's what I think. You cannot simply rely, well, this is from where? Paraguay or Chile?
If there is proof, I mean, even if the agroecological zone is similar, I cannot use data. I have to.
It's similar, but it's not never again. It's not the same. It's maybe similar, but similar to what extent? I mean.
It cannot be. It cannot. Okay, I have to.
You cannot simply use data from Paraguay only. That's what I'm saying.
So he has to do all these tests like.
But that's what I'm saying. Maybe not all the stuff, but at least maybe some limited field trials to prove his point. You see?
Okay.
John.
Just to react to that a bit, one issue is, statisticians will argue sometimes that you can get sufficient statistical power in a compositional assay with, 4 sites with some replications within, rather than the 8 EFSA calls for. But at a level of 4 sites and the usual array of metabolites you measure, that's in excess of $100,000 to get that work done. So if you up to 8, you're at 200,000, and if you do 8 commercial varieties around there, 200,000 times 8 is $1.6 million.
And that starts to get out of reach for some of the public sector projects. Okay? It can't do that, and that's where referring to these databases is really helpful and effective. And I certainly take your point that you'll see databases, the most rigorous, OECD is very, very good, and there are many more samples in it. Okay? So I think some of the noise, potential noise that you might get, because they might not be using standardized methods, or doing it under GLP, for example, that probably gets swamped out by the greater breadth of samples and environments. So, my feeling is that's a valid way to proceed, but often, the only way to proceed.
But yeah, no, I agree with you. It would be very good if we, at an international level, could come to some kind of agreement on what would be minimal requirements for a field trial to perform.
Yeah.
I mean, if you talk to statisticians at EFSA, I'm not saying they are going to kill you, but these are statistics and statistical experts, and everybody has it, not everybody, but there are certain groups who think, well, we should do this as a minimum. But apparently, at an international level, we could do better for sure. But I've always been advocating that OECD, and especially in the area of bioethics, should do more instead of having these meetings of the task force. They should look at their consensus documents, because there's a lot of flawed data in the consensus document when it comes to natural variations of compounds.
So maybe that speaks to how the consensus documents are worked on or are developed. Because for me, the consensus document speaks to the kind of knowledge and expertise that they have at a particular time, because they are involving a certain number of experts who have. So that is what we know at that time. But as we speak now, they are going to revise the consensus document for maize. That's because it's recognized that we probably know more about maize now than we did in 1996 or the year 2000 when that document was developed. There's new information that can be added, and so it's going to be revised.
Yeah.
And so from time to time, those documents are revised, but it also speaks to the kind of aspect that the various delegations put together to actually develop the documents, and the extent of consultation that goes into finalizing it and declassifying various documents. Yeah. But yeah, OECD can do more. I agree with you. Yes, please.
Thank you for your presentation. I'm curious about your position on the use of microbial express proteins for toxicological studies.
Well, we do it a lot. I mean, in the dossiers we are getting, we have these proteins derived from microorganisms. The key thing is that you have to prove equivalence with the protein being expressed in your GM plant. So there are a number of biochemical techniques which you should use and compare your protein from your microbe with the one expressed in your plant. So it's fairly easy standard stuff to do for an applicant. So that works quite well.
Are you asking if it's a good thing to do, or?
No, I mean, of course, proteins that are expressed in plants are different from maybe slightly different from what is expressed through a microbial system.
Yeah, it could be. Yeah, it happens.
Microbial express proteins.
It happens, but yeah.
So
and what are the modifications? Is it glycosylation or?
Yes.
Or is it? But also in terms of amino acid sequence, slight differences.
There could be differences.
Yeah.
But the question is, what's What's the alternative?
There are no alternatives. You mean
Because some of these proteins are expressed at such low quantities.
you have to do it, yeah.
You need truckloads of elephants. the, the, I tried to estimate how
How
much,
many?
what hectarage of maize field we need to cultivate to be able to get gram quantities of fry protein in a particular event.
Forget it.
It's not possible.
No.
It's just not possible. It's just not possible. And to think that you need quite, because you need to feed the rats very high dose to be able to do these acute toxicity studies. And for you to cultivate that amount of maize and just to extract this amount of DNA to, protein to, it's not practical. But anyway, I'm sure we would. You
But it's an essential point of the dossier, yeah? What you-
tomorrow, yeah. Okay, any other question, Kwame?
Follow up from the earlier presentation. In the light of when we look at regulatory endpoints, considering Europe doing regulation from the point of the process for making the crop, and let's say Canada looking at it from, as a novel food, are there any differences?
But I think you are less worried about definitions on genetic modifications. Is that a genetic modification which falls within the definition? Yes or no? Same thing for transgenesis and talents and so. So that's a discussion. Yeah, but in practice, I mean, Canada primarily focuses on the characteristics of the GM food.